Welcome to the Inner Universe, a journey into the cells of the brain. In this video, we will explore the structure and function of neurons and glia, the fundamental building blocks of thought and consciousness. The brain features two main cellular protagonists, neurons and glia. The human brain contains approximately 85 billion of each. While roughly equal in number, they perform distinct and complementary roles. Neurons are the performers. They sense environmental changes, communicate with other neurons, and command the body's responses. In contrast, glia act as the support crew. They contribute by insulating, supporting, and nourishing neighboring neurons. To visualize this relationship, if the brain were a chocolate chip cookie, the neurons would be the chocolate chips, while the glia would be the dough, filling the space and suspending the chips in their appropriate locations. Next, we examine the anatomy of a neuron, which represents the architecture of information. The neuron, or nerve cell, serves as the fundamental unit of information processing in the brain. Its structure is elegantly specialized for communication, consisting of three basic components. First is the soma, the central cell body. Second are the dendrites, acting as the receiving antenna of the neuron. And third is the axon, the transmission cable that sends signals away from the soma. It is important to note that collectively the dendrites and axon are referred to as neurites. We now enter the command center, a look inside the soma. Also known as the cell body, the soma is the neuron's metabolic heart, typically measuring about 20 micrometers in diameter. It is enclosed by the neuronal membrane, a 5-nanometer thick barrier that acts as a gatekeeper, controlling what enters and leaves via specific pumps and channels. Internally, the cytoskeleton provides scaffolding to maintain the neuron's shape, while organelles like the rough ER and Golgi apparatus handle protein synthesis. Finally, the mitochondria serve as the essential powerhouses of the cell. Next, we examine ATP, the energy currency that fuels the brain. Neuronal communication is an energy-intensive process driven by the mitochondria, small organelles about 1 micrometer long. They generate adenosine triphosphate, or ATP. This chemical energy is used to power special active pumps within the neuronal membrane. By doing so, these pumps establish critical ion concentration differences between the inside and outside of the cell, creating the electrical potential necessary for sending signals. Now we move to the axon, the brain's information superhighway. Unique to neurons, this structure acts like a telegraph wire, specialized for transferring information over distances. It originates at a region called the axon hillock. By the numbers, axons are impressive. In humans, they can extend up to a meter or more in length. Crucially, diameter dictates speed. As the comparison between a human axon and a squid giant axon illustrates, the thicker the axon, the faster the electrical impulse travels. We arrive at the point of contact, the anatomy of the synapse. At the end of the axon sits the axon terminal, or terminal bouton. This is the junction where the neuron passes information to other cells. The structure is divided into two sides separated by a gap called the synaptic cleft. The presynaptic side consists of the axon terminal, while the postsynaptic side is usually the dendrite or soma of the receiving neuron. The transfer of information across this gap is essentially what we call synaptic transmission. Inside the axon terminal, we find a specialized environment uniquely structured for communication. It differs from the rest of the axon in 4 key ways. First, the internal scaffolding of microtubules does not extend into the terminal. Second, the terminal is packed with synaptic vesicles, small membrane bubbles that store chemical messengers. Third, the inside surface is lined with dense proteins, which serve as the machinery for release. And fourth, the area contains numerous mitochondria, highlighting the significant local energy demand required for synaptic transmission. This process is known as the handshake, where the synapse translates an electrical signal into a chemical one. It unfolds in 4 distinct stages. First is the arrival. An electrical impulse travels down the axon to the terminal. Second is the conversion to chemical. This impulse triggers the release of a neurotransmitter into the synaptic cleft. Third is crossing the cleft. The neurotransmitter diffuses across the gap. And finally, the conversion to electrical. The neurotransmitter binds to receptors, converting the chemical signal back into a signal in the next cell. To maintain function, the neuron relies on a sophisticated logistics network known as axonal transport. Since the axon requires a constant supply of materials from the soma, it utilizes the microtubule rails of the cytoskeleton to move cargo. The primary method is antegrade transport, which moves material from the soma down to the axon terminal. This process is powered by the protein kinesin, which literally walks vesicles along the microtubules, consuming ATP for every step. Conversely, retrograde transport provides a separate mechanism for moving material in the reverse direction, sending signals from the terminal back to the soma. Now we turn our attention to the dendrites, the antenna of the neuron. While the axon speaks, the dendrites listen. Their primary function is to receive incoming signals from other neurons. Acting as receiving antennae, they are covered with thousands of synapses. Specifically, the dendritic membrane located under a synapse is rich with specialized protein molecules called receptors. These receptors are engineered to detect specific neurotransmitters released into the synaptic cleft, essentially initiating a new signal in the receiving neuron. Finally, we meet the unsung heroes, the glia. Once thought to be mere passive support, glial cells are now understood to be active and essential partners in brain function. They are not just the dough, but the bakers that maintain the perfect environment for neurons to work. The most numerous type is the astrocyte. Filling most of the space between neurons, astrocytes play an essential role by actively regulating the chemical content of the extracellular space. For example, they strictly control the concentration of potassium ions in the fluids surrounding neurons, a process that is critical for proper signaling. Next, we look at myelinating glia, which serve as the axon's insulators. Oligodendroglial and Schwann cells provide layers of membrane called myelin. Much like the plastic coating on an electrical wire, this sheath speeds up the propagation of nerve impulses. A critical feature here is the node of Ranvier. These are periodic gaps where the axonal membrane is exposed, which are essential for high-speed signal conduction. There are key differences between the two types. Oligodendroglia are found only in the central nervous system, and a single cell contributes myelin to several axons. In contrast, Schwann cells are found in the peripheral nervous system, where each cell myelinates only a single axon. We conclude our journey with this perspective. The brain has been called the most complex piece of matter in the universe. By exploring the intricate machinery of neurons and glia, we begin to appreciate the massive scale of that complexity operating within us every single moment.